All lab tables will be disinfected by spraying with 70% alcohol before and after any lab work. Paper towels that we use can go into regular trash. Hands should be washed before and especially after lab work. Paper towels that were used for drying your hands can go into regular trash. No food or drinks are allowed in the lab. Long, fabulous hair must be tied back. Shoes must cover entire foot. No bare shoulders. You can wear a shirt, sweater, or apron to cover your shoulders. Eye protection like goggles must be worn, and a disposable apron or lab coat. Goggles provided by us are sterilized in the UV cabinet and must be returned to a UV cabinet after every lab. Each student has 7 items for use during the semester: rinsing tray, rinsing bottle, inoculating loop, ruler, slide boxes, Sharpie, and a clothespin. Take out the slide box with your instructor's name and write your name on it. Slides and slide boxes may not be taken out from the lab under any circumstances. Stain bottles are provided in a tray by the sink. All staining must be done in the staining tray and chemical hood for special stains. Heat-fixed and stained slides are not contaminated. Uncontaminated slides for disposal can go into the cardboard box. You may not take prepared slides out of the lab to study in a different location. Wet-mount slides have live organisms on them. Do not put these slides in your bag or take them outside to study. Wet-mount slides must be placed into a dish with disinfectant for disposal. Each student will be assigned a microscope and have a numbered space in the cabinet. Your desk number corresponds to the microscope number. You're responsible for taking care of your microscope and reporting any problem with it. Kim wipes used for cleaning lenses are discarded in regular trash cans. Before putting the microscope back into the cabinet, you must wipe off the lenses, set the objective lenses to 4x, clean the general stains on the microscope, and wrap the cord properly. Hazardous materials in the lab include open gas jets, hot micro-incinerator, chemicals, live culture, reaching across the table, not paying attention to your surroundings, and glass slides and red biohazard bag. Although the bacteria which we'll be working with are not considered to be pathogenic to healthy humans, always assume that they are infectious and act accordingly. Never ingest or smell bacterial cultures. Cultures provided for lab exercises are found on the site counter. The test tube racks are color-coded for each table. At the end of each lab, collect all the culture tubes and plates, place them on the labeled rack, and return them to the site counter. If your group has more than one Petri plate, stack them on top of each other, wrap the stack with one piece of tape. Then write your group name on the tape and place them in a plastic box on the site counter. For tubes of media, label the tape around the tube just below the cap or use a marker to label directly on the tube. Combine tubes in the tester racks for incubation. All rinsing and decolorizing must be done on a staining tray. Liquid waste from staining in your staining tray must be poured into the jug with a funnel by the sink. Do not pour it into the sink. Discarded Petri dishes after recording the results. Used swabs and microfuge tubes go into the red biohazard bags on the tables. Discarded cultured tubes, which are not broken, are collected in wired baskets on a card in the lab. Take the labeled tapes off the tubes before placing them onto the card. Used plastic pipettes go to round plastic jars on tables, and the packaging goes into the regular trash can. Immediately report any broken glass, test tube, or accidents to your instructor. Any plain working glass without bacteria, such as glass tubes or slides, may be put into a cardboard box. If you break glass tubes or slides with bacteria, the area must be saturated with disinfectant. Paper towels used for cleaning bacterial spills is contaminated and must go into the red biohazard pack immediately. Broken glass must be stripped up and put into the metal canister for sterilization. Never put glass or anything sharp in the red-orange autoclave bags.